Hello, in this video we will discuss about the lagging strand and leading strand and during the DNA replication of eukaryotic DNA replication. Eu mean true carrion mean nucleus while the prokaryotes is the bacteria. Pro mean before and carrion mean nucleus. So no nucleus are present in the prokaryote. But eukaryote during the DNA replication we will learn and we will just understand about the lagging strand and leading strand. So this is the cell, eukaryotic cell, contain chromosome within the nucleus and cytoplasm and the plasma membrane. So the DNA contain chromosome contain histone protein and DNA thread, anti-parallel, double-stranded anti-parallel strand, 3' to 5' direction, one strand, and other is the anti-parallel 5' to 3'. So this is the topoisomerase enzyme used. This topoisomerase enzyme used for the enable DNA to unwind during the growth factor or the cell division signal. So in this way, here you can see this is the leading strand and this is the lagging strand. Leading strand means it is 5' to 3' direction, while the 3' to 5' direction is the lagging strand. Lagging strand is a problem, but while the leading strand is nothing. So here is a sliding clamp will be attached, while the single stranded binding protein also attached here. Single-stranded binding protein is used for the prevention of unwinding, and this is the replicating fork will be developed. This replicating fork is used for the replicating fork used toward leading strand toward the replication fork synthesis of the another strand, while the lagging strand is used for the away from the replicating fork, the synthesis of new DNA. So this is a CMG helicase enzyme used to unwind the helical structure, while this is the polymerase epsilon, is the primary leading strand polymerase. In this sphere is the nucleotides addition. This nucleotide addition site is the hydroxyl group easy, and the leading strand is easy to synthesize, while the lagging strand is the problem. Problem for what? Problem is the hydroxyl group are present in the 3' side are not present because its synthesis through 5' to 3' direction. So the primase enzyme creates RNA primer will first bind because the 5' to 3' direction synthesis is important. So the primer contain 3' side is hydroxyl group. Hydroxyl group is important for the synthesis after the binding with the polymerase enzyme. After the primase release, after the binding with the primer. So here is the polymerase alpha. Synthesis first stretch of DNA after primer. So after this, the single-stranded binding protein is here, and you can understand this polymerase alpha synthesis first stretch of DNA after primer, and again primer addition with the segment wise. So again the next primer will attach because the 5' to 3' direction is very important because the 3' contain hydroxyl group is required for the synthesis of new nucleotide with binding through polymerase enzyme. So remember, this is lagging strand is going away from the replicating fork, while the leading strand is easy and it is simple and go toward the replicating fork. So this is a single sliding clamp are also attached will lead to polymerase delta is used for the primary lagging strand polymerase for polymerization to addition of nucleotide. After this, the primer is left behind. This primer is synthesis for the RNA, so it should be removed. After the removing of the primer, the Okazaki fragment will be left behind. This Okazaki fragment is contain 10 to 100 base pairs containing Okazaki fragment. It will be removed through a exonuclease remove RNA primer. After the removing, the segment is the gap are present which that will basically is the Okazaki fragments. And in this way the Okazaki fragments will be ligase rejoins strand, and in this way the ligase enzyme used to synthesize the gap, the Okazaki fragments. After this, the ligase enzyme will be attached. The polymerase also, the primer, which that is where the primer side will be filled. The ligase enzyme will ligation of the strand, so the segment will be covered. So in this way, here is the away from the replicating fork, the synthesis after the releasing of the polymerase delta primer lagging strand, the polymerase and the ligase rejoin strand. And in this way, here is the primer site and here is the primer site. So the exonuclease enzyme used to remove this primer to addition of this. This is the nucleotide, and to complete a synthesis of the lagging strand to complete the replication in this manner. So remember, the DNA polymerase catalyzes the addition of nucleotide at the 3' hydroxyl group. And in this way, the 5', basically the 3' is requirement for the hydroxyl group for the addition of the further nucleotides. So this is the 5' to 3' direction. So the other strand is the 3' to 5' direction is very important. In this way, the primer are basically primer, you should need to understand. Here is hydroxyl group, and this hydroxyl group is used for the addition of nucleotides, and in this way the addition of nucleotides is important. While on the other hand, here is the hydroxyl group requirement through 3' side provided by the primer, and the providing of the primer due to the hydroxyl group here and here. You can understand I am drawing here. So remember that things. This is the hydroxyl group is important for the synthesis of the further nucleotides. So after this, nucleotide will be removed, and in this way you can see here is the DNA polymerase catalyzes and addition of the nucleotides at the 3' hydroxyl site, and in this way the providing of the primer is very important for the binding site for the 3' side to 5' side. Every time the 3' side to 5' side is the synthesis of the DNA. So the primer is very important for the synthesis. So this is a single cell replication required DNA replication first to double. After this is known as the karyokinesis. After the karyokinesis, the cytokinesis will occur. The cytokinesis means the full cell divide after the replication of the DNA. And before the replication, the mismatch check to clear the DNA problems and the prevention of cancer, and the epigenetic homeostasis due to the methylation homeostasis and acetylation homeostasis will be checked. And in this way, the epigenetic mutation prevention. On the other hand, the prophase and metaphase, anaphase, telophase, cytokinesis, and the karyokinesis to cytokinesis will occur. So let's begin to understand the here quick overview. This is the prophase. We'll go into the metaphase. Prophase means the preparation, and the metaphase means the metaphase plate will be formed, and anaphase means the chromosome will be separate after the replication of the DNA, and telophase is the nucleus will be divided. Two nucleus will be formed, is known as karyokinesis and cytokinesis. Thanks for watching.